गुड, कितना अच्छा लग रहा है, लोग आए हुए हैं, राइट? ये सब यहाँ कहाँ गए? आई थिंक फिर भी अबाउट 30% आर एट होम, राइट? 40% पूरा बैच में, राइट? सब घर गए हुए हैं ना? अच्छा, ओके, सो आई थिंक because now you are starting the project, and I think you are looking at a couple of papers. So you do require some background in biology, right? So that you can better understand them. So I'm going to cover certain basic things which are very important. Of course, biology is very beautiful and very large. I mean, very vast, right? So you don't have to look at everything, but certain basic things, basic understanding is very important. And there is a very nice course by Eric Lander. If you want to look at, that course name is Secret of Life, available on EDX. Okay, which I think everyone, each of you should watch, even if you are from computer science, because it's a very important course. Eric Lander is a mathematician and also professor of computer science and biology at MIT. Okay, he's also founder of Broad Institute, and because of him, a major change has happened in the area of biology since 1989 or so. Okay, so I think you should look at that course. I mean, you should register for that course. Of course, it is freely available on EDX. But if you want certificate, there is some waiver program which you can apply and so on and so forth, right? So you know about it. How to do a course on EDX. In my opinion, every high school student should do that course. And believe me, in class 11th, you can do it. So you can tell your brothers, sisters, and whosoever that to do that course. Biology contains all your ICT. Okay, so your entire ICT is subset of biology. Okay, so it is very important that you should do the course. Unfortunately, in India, when at 11th or 10th, maybe you have to choose bio stream, or though I have been speaking at many government places about this, and it's very difficult to change, but I think some boards have allowed it to opt, right? And the new education policy actually allows that, but slowly it will be changing it. So as a computer scientist, biology is must for you. You can see that because this course is about DNA storage, right, which is involving biology. The computer heart of a storage is actually computer science. Is the storage is the heart of computer science, right? We cannot do anything without a storage, and you know, biology comes there also. As I mentioned, this is very important code that entire ICT that you study, each and every concept is already there in biology, even much more what you can think of. Okay, so if you really want to understand the nature or any concept in ICT. In fact, almost all good machine learning algorithms came from biology. Okay, that you do all AI and everything. So for example, neural network is one such nice example. Okay, so many interesting algorithms are there, inspiration from nature or biology, right? So it's very important. So you should have a good understanding of biology, basic things. So I think in your class 10th, what biology you might be studying was probably collecting the plants, flowers in the book, something like a collection and so on and so forth, right? But that's old biology. So modern biology is more about molecular biology, right? So it's something very important. And there are some brief books also, like biologist, molecular biology for computer scientists, very small book available, you can look at it. But I think that is also a very elementary book. The discovery of DNA computing happened because Edelman, who was one of the person in RSA crypto system, right? When he was reading this biology book by Watson, right? And he realized that, well, all these biological things can compute. The DNA can compute. And then he designed the first experiment where he actually solved a traveling salesman problem, an instance of traveling salesman problem with a bunch of DNA molecules. And I think I sent you a link yesterday about something. A circuit has been made, just like that FPGA people have made now. DNA, programmable gate array, right? Which can really solve square root and equation solving through bunch of DNA molecules. All these DNA molecules, the DNA circuits works like this. There are a lot of DNA strands interacting with each other and solving the problem for you, like fighting and then giving you the answers. So it's really beautiful. Of course, there are some simulators also for doing these kind of things. Okay. So, actually, this biology changes something around late 80s, 85, 86. What happened? This Eric Lander actually did his PhD in coding theory, correcting codes. And being a mathematician, he thought that what to do next. So not sure. So he actually started teaching mathematics in management department. I think it was Harvard, if I'm not wrong, but that's his starting point. And one day, actually, at a dinner, I think his sister was there who was asking what you were doing or something. A friend, I don't remember the exact who was there, but anyway. So they asked what you are doing. So he said that, well, I have done PhD in error correcting codes, which is all about information processing, right? How much information you can compress, how much information you can transmit, and errors will happen, how to correct the errors, and so on and so forth. So that person said that, well, inside our brain, so the expertise of the person was in biology only, that inside our brain, there are a lot of information processing going on. Maybe you should look at that problem. Okay, so just by curiosity, he started reading all these biology books and so on. Slowly, he realized that if you want to understand brain processing, you need to understand genetics. In genetics, uh, what was happening at that point of time? People were reading genomes. What are the genomes? The content, I mean, the DNA that is inside each of our cell. For every living thing, there is a genome, right? So for you, for any plant or whatever. So this genome contains instructions for making whole bunch of you, who you are. And at that point of time, you see, most of the biologists, they were, suppose you want to read some genome, right? So even a small genome. So initially they tried to read some bacterial genome, so bacteria and so on, small things, and that used to take a lot of time for them. Even if the size is not too big, it used to take a lot of time. Eric Lander realized that, well, if we really want to understand biology, we need to look at all this genome, right? And especially for the human genome. So human genome estimate was that there are 3 billion base pair, 3 into 10 raised power 8 ACGT. That is the size. Okay, there are 22 chromosomes, basic ones, and then there is another one. So these chromosomes, so are very, very important. Okay, so we should somehow read them before understanding what they are doing, right? We need to read what are these sequences, okay, and then which portion is giving us what kind of protein, because normally the human genome, which is about 3 billion base pair, only 2% codes for protein. So our body is made of protein, everything like your eyes, hair, nails, everything, it's made of protein. Protein is a building block of our human body or any other thing, right? So instructions for making this protein in the human genome is only 2% out of whole 3 billion base pair. Rest is all control system sitting there, which is telling you that, okay, if you are feeling hungry, which protein should you produce, right? Or any biological process, some protein has to be produced. Somebody will go there, read the genome, a portion of DNA from one place, another portion of DNA from other place, splice them, and give rise to particular protein that you want. In what amount, how much protein has to be produced, everything is controlled. So 98% is controlled. And despite of so many efforts since last so many years, still we understand very little about it. Of course, people are trying to understand, many things have been done. A lot of progress has happened because of this sequencing business, right? Reading the genome. So initially, when Eric Lander wrote this grant to National Institute of Health, NIH in US, that we should study the human genome, biologists said that this guy is mad, We are only trying to study a small portion of bacteria, and it is taking us so many days, maybe month or two. And this says that I want to read the whole 3 billion base pair. This is not possible. So the reason why it was not possible, because the sequencing methods, reading the genome, used to take a lot more manual operations. Okay, you cut the genome into specific pieces, and then you do the PCR. PCR is a procedure which will do the replication of the DNA, right? then you read them, then somehow you reconstruct those pieces into the bigger genome, and then you see what is that actually ACGT stands for, right? So ACGT genome is nothing but a specific order of ACGT, adenine, cytosine, guanine, thymine. A specific order will give you some particular property. Okay, so this is something very, very important. Scientists also realize that a lot of disease that happens in human body is because of error in the genome. For example, uh, at chromosome 7, in the P region, there are two regions, P region, Q region, any chromosome. The only deletion of 3 letters of ACGT will give you a disease related to, your lungs, which is known as cystic fibrosis. Okay. So these deletions of ACGT while doing the things, right, can give rise to different kind of disease. So obviously that means people want to study the whole chart, right, of the human genome and try to map what kind of things are there, how they are related, which deletion will give you what kind of disease is an important thing, right? Anyway, so what happened at that point of time, I think this was around 80s, 85, 80, 88, 89. So finally, actually, Eric Lander, then you see, he was a great mathematician. I mean, he's a great mathematician anyway and biologist now. So he actually somehow convinced, the biologists that we should go for it. Then finally, Human Genome Project was conceived, okay, approved by the US government, and which involved many, I think, countries that we should chart the human genome and read the whole genome, right? And, you know, because of that, once this approval was there, that means funding is there, people want to now study, right? Suddenly there was, a race for building, reading the whole genome, and then maybe try to come up with some different kind of solutions that can be done, right? So for this disease, you should do that. And I think some of you might be knowing there is a company in India. Of course, there is also one company in US, 23andMe, which was started by the wife of Sergey Brin, the founder of Google, right? So they wanted to study the relationship of different diseases and so on, right? So they do that kind of thing. So if you give your saliva or anything to them, they will extract the DNA and they will read it, and they will say that this guy will have chances of having this disease because of, genome has these kind of things, right? And this Anu Acharya in Hyderabad, she is having this company. So they have come up with a product known as Genome Patri, just like Janam Patri. They have Genome Patri, where you can match genome before marriage. So this service is available. So you know that, okay, which your offspring, if you want to minimize certain disease, right? So you can actually match Janam Patri ke saath saath Genome Patri milana bhi zaruri hai right now. Okay, because then only the chances of disease will be less. So this service is available, by the way, in Hyderabad. Okay, it's not a very costly thing. So these kind of services are possible because now we are trying to understand more and more things. At the same time, what happened? So I still remember when one particular chromosome was read, I got that special issue of Nature actually at my desk. In fact, I was doing PhD at that part of time at IIT Kanpur, and I think it was 95, I guess, when they read one particular chromosome. Okay. And complete chromosome was published. And later on, I think this Human Genome Project was finished in year 2003 when they read the entire thing. Okay. And then once you read human, you should read cow also, you should read dog also, everything. Suddenly, a lot of money is pumped by many governments all over the world. Okay, let us read everything. Right. Read and make database. And then the huge database is created at NCBI, which is part of National Institute of Health in US. So this US government controls all, of course, there's a consortium also many. So each of the country contributes new sequences after reading the data, okay, into the NCBI database. Okay, I can show you NCBI database right away. Right. So people started depositing these genomes and trying to understand it, right? Try to make some sense out of it. In fact, we have also developed a software, biospectrogram, which can do the spectral analysis of genome. For example, you want to know gene, where is the gene? So as I mentioned, out of total human genome, only 2% codes for protein. So how do you protein? How do you know? So whatever codes for protein is known as gene. How do you identify which portion will code for protein? So actually, what you can do, it's a really beautiful thing. People have found that You just map ACGT because it's a symbol of ACGT, right? You map this to some numerical values. Could be, for example, if there is a place 1, otherwise 0. You will get one characteristic sequence of 0 and 1 binary sequence. Similarly, so this is known as indicator sequence for A. Similarly, you can have indicator sequence for C, indicator sequence for G, and indicator sequence for T. Right. So these binary sequence you can see wherever A is coming, you place 1, otherwise 0. Right. Wherever C is coming, you place 1, otherwise 0. So you get binary strings. And from that, you apply transformation, Fourier transform, and you look at the power spectrum. And if you look at the power spectrum, so basically what scientists do, they look at the size of window and then they move along the genome. Wherever there is a peak observed in the power spectrum, more likely there is a gene coding. No, instruction for protein is there. All these kind of techniques have been developed. A lot of softwares have been developed by so many people. And that is what whole area that is known as bioinformatics. Bioinformatics means developing tools for whatever data has been generated by sequencing, means reading all the genomes and then managing them and developing software tools which can actually analyze that data, okay, for different disease and other things. It's still, so this data is all open, available to all of you. If you want, you can form a company, do a specific thing and actually do whatever you want with this data, right? Plenty of these, just like the Genome Patri service is there. Maybe your company may have much better service, right? So they will say that, okay, I will avoid this disease number, this, right? You say that, okay, in my software company will give you much better solution. You can avoid these diseases in your offspring, right? Not only that, now this has gone into even a couple of steps ahead. Now, what people have, what is known as synthetic biology. There is again this guy who started DNA storage, George Church. He said that let us stretch genome on my own. Take the instructions and write the genome. In fact, people have done the experiment. They have taken one bacteria, so genome of one bacteria, and they know the genome of another bacteria. They make changes in this genome of string of ACGT. Of course, program is required, and then one species transformed to the other species. So these kind of things are possible, right? So transformation between different life forms is possible. They have done the experiments. Okay, you see the kind of power you are getting in your hand, right? That is there in this synthetic biology. People want to grow plants which can glow in the night. How do you do that? They looked at certain species which actually give light, right? Take that gene, insert it here, right, and do the things, right? And then they have observed that you can do that, right? There is a fish, zebra fish inside the ocean. If you cut the fin, actually, it will regrow automatically. They have identified which gene is responsible for it and insert it in a hen, and they were able to regrow the leg of the hen, right? These kind of things are possible. All kind of experiments are possible, right? This is all what? Computer science, biology, all interdisciplinary thing, right? You require software programming, we should look at the data, do the analysis, and then you do the experiment, right? And you grow it in the lab. Those kind of things are possible. You see, the biology is really very, very powerful tool now. So synthetic biology has evolved over the years in a lot of things. Now, China is trying to create designer babies. But of course, there are certain regulatory approvals is required. Not every government is allowing this. And so you want children who have high IQ. So you can do that by modifying the genes, right? Or if you want a person with 4 eyes, you can do that. All those things are possible in practical, in theory, basically. Of course, practical things will require approval from different government, right? But do you really want to see those kind of creatures on the planet or not, right? This is the unlimited potential. Yes, any question? Yes. Huh? Mm-hmm. Yeah. So in fact, there are experiments that like there was one lady, she was sort of blind. And the reason they found out that there was a particular protein which was there in excess inside the eyes. So what they did, they switched off that gene. So the protein formation stopped and she started visualizing the things. So these kind of things are possible, right? Experiment will of course involve cost, right? So that's why you see software and simulations are required so that you can actually do the things before doing the experiment. Experiment will involve, the money will be there, right? But you see, the opportunities are quite a lot for you, right? I mean, you can build a company, Data is available free, right? Take the data, play with it, right? And actually, you can write all kind of things and different kind of things. People are doing all kind of things actually in this area, right? All kind of things. Uh, you know, as I mentioned, so these trees also they have demonstrated. They have taken a bacteria, they have created what is known as bacterial bulb. Okay, so the input is bacteria which will automatically grow in the night. Right? So they have actually in UK on one road they have actually just put all these bacteria in glass chamber and everything, and they glow in the night. So there is no electricity required, right? So all kind of things are possible. So when you play with biology, so biology is at that. That's why I say that probably now you will also agree with me that biology has unlimited potential, right? Not the biology what you have been taught, right? So plenty of opportunities are there, right? All kind of things are there, right? So also people are looking at medicinal, all these plants and so on, genome them, what are the properties they have and so on, right? Luckily, I saw government of India also started a lot of things. In fact, in Gujarat, there is Gujarat State Biotechnology Mission, GSBTM, which was started something around, I think, 2001 or 2 or something like that. And they have also done a lot of things. Because see, as I mentioned, this started in 89. Just because of this Eric Lander, the whole thing is started in a very, very, uh, you know, aggressive manner by so many governments. And in fact, there was one person, Craig Venter, who was in his team. He left the team. He started his own company. Right? I mean, this kind of things will happen. And they are competing. And now many companies are there, right? So as I mentioned, now the kind of things that are possible. Now there are governments who want to control. So now, so you see, if you want, as I mentioned, there are some people who have taken a computer virus that you have, that infects some, you know, malware or something. Take that, they have taken the virus, they have converted into ACGT, right? And those ACGTs basically synthesized by synthesizer, DNA synthesizer, and you ship it to someone. Person will, they have read it back, and that becomes actually uh, infected file which infected the whole computer system. That they have done actually successfully. So DNA malware attack, that is possible. And you see, in terms of our DNA storage, when you are storing data on DNA, what kind of things are possible, right? People will insert something in between, right? All kind of things are possible, and then you have to make, you have to protect your data in some way. So security is also a major issue. In fact, since all these things are controlled by synthesizer, so DNA synthesizer, now I think US government want, I think they have made an international thing also, that whatever you are printing, you are not allowed to print anything. It has to be approved by someone, right? Maybe you want to maybe you want to make a new kind of virus. Corona you have seen, right? So if you want new kind of, so people are trying different kind of experiments, right? And I think some people say that it is because of these experiments Corona actually came, because they were making changes. Can you make more deadly virus? All kind of things are possible, right? Playing with things. Playing with virus means microbes. Virus and bacteria is quite easy. Small genome, right? Playing with large things are a bit tricky thing, because that takes a lot more time and efforts and so on and so forth. Right? So most of these biology labs have certain approvals level 0, level 1, level 2, right? And some of these labs can only do experiment of certain level. So basically what I'm saying, you are not allowed to अपने घर में जाकर के experiment करना शुरू कर दिया, right? So basically there is a limit. I mean, you have to take approval from the government. Government should know what you are doing, right? So that is the thing, because you may do dangerous things as well. All kind of people are there in the society. By the way, uh, as an exercise, any of you want to see your own DNA? You can see that. Each of you at home. Very simple experiment you need to do. Or if you want, you can look at strawberry. Strawberry, many common. It's quite common. Or you can actually look at this banana or anything, any fruit. What you require? Detergent, small of alcohol, that rubbing alcohol, right? And on YouTube you can get thousands of videos how to extract DNA at home. Doesn't require a lot of. You can do it at hostel also. Okay, rubbing alcohol, you spit. You have it at home. detergent, soap, you have it, right? To break the DNA, all those walls and everything. And still it, mixie is also available, right? Or you can also do it by hand also, that's not a big deal. So you can actually see the DNA. So I give you in this experiment, you should actually see actual DNA. Okay. This experiment you should do. Okay. Then you see. Now the question is, we want to read this DNA. DNA you can see, but how do we read it? Okay. So that requires different techniques. So as I said, so you should know chemistry, physics, and biology, of course, mathematics, computer science, and VLSI and semiconductor. सब कुछ चाहिए. You have to make a product, right, which can read the data. Okay. So as I mentioned, sequencing has evolved quite a lot because people wanted to read a lot of genome, so they developed different sequencing techniques. So recently came up with next generation sequencing. So normally, how do they read? It's very. I'll give you this gist of this. You take the genome, cut it into pieces. Okay, it's like that. You want to read a book, first tear the pages, make photocopy of it, and then reassemble these pages by sophisticated algorithms, which will do the reconstruction of the whole file or whole book again, and then you read it. So basically, you cut it into pieces, do the PCR polymerase chain reaction. It's very powerful. The person who discovered it, he got Nobel Prize. Very simple technique to make copies of, you know, portion of DNA, whatever you want to replicate. And so basically, because when you are doing some experiment, if you have a small portion of DNA, you may not be able to, I mean, it may be not enough for you to do the experiment. So you replicate, you have enough copy, you take some problem is there, you can take another copy and so on. So PCR is done all the time. Okay. Even in the DNA storage, people do PCR. I think some of you might have read those things, right? I'll talk about PCR also, polymerase chain reaction. There is a, I think at Georgia Tech, there is a virtual lab where you can actually do the experiment from your laptop. Okay. So you can actually do the PCR. You can also actually do the all other like gel electrophoresis. Gel electrophoresis means you measure the length of the DNA, what you are actually looking at, right? Those kind of things are done. So these are the remarkable things actually. And biology has changed quite a lot because of PCR. All these tests, PCR is very common technique. Okay. Yes. Okay. So I think uh, so some of these techniques I'm going to now tell you exactly how things happen briefly. Okay. And then we'll go about some specific details also, right? Now people have developed some specific methods for cutting DNA. Okay. Copy, as I mentioned to you, or, even joining them by ligase, which is a protein, which does a lot of things. So especially if you want to be an expert in DNA storage technology, you need to understand biology really well, right? How things are happening. Okay. Because otherwise it will be difficult for you to understand the whole concept. And this is a challenge because most of the data storage companies, they right now do not have enough experts. They are only IT people, right? They don't know biology, they don't know chemistry. But actually, in the next, as people are predicting by 2030, we will have these kind of data centers replacing cloud with the DNA storage thing. Okay. So let me show you. Okay. So 1665 is the year. First time Robert Hooke observed, that microscopic biology was discovered. How many of you have seen the cell and everything by microscope? Any of you? Very good. Large number of people. Each of you should have a microscope at home and see bacteria and everything, right? Okay, in the curd and so on, you've heard. So observation that organisms are made of cells and so on. By the way, 1865 was very, very important year. Mendel experiment, you might have heard about it in biology, right? And it happened because of history of ships. I will talk about that. Okay. 1869, John actually discovered DNA, named it nucleon. Then from 1880 to 1900, now you see, when there was industrial revolution in Europe, biology nobody was still knowing much. You see, not much progress. That's why everybody is saying now this century is only for biology. Forget all other subjects. Internet has shown you a deep impact on everybody's life for the last how many years? 20, 30 years, right? And still you are looking at all the other kind of things, right? It has created so many. So the BTech in ICT program is created because of discovery of internet only, right? And similarly, the biology is going to shape the next future. Everybody will be only doing biology. Okay, of course, but all other things will be there. So chromosome, was observed in 1881. And finally, in 1899, Altman renamed nuclein into nucleic acid. All this I have taken from this website actually. And I have actually placed a PowerPoint from this website in the lecture folder. You can see in more detail if you want, right? Because it's very large slides. Anyway, now amino acids are very, very important. Proteins are made of amino acids. Okay. So they have a specific structure. There are different kind of amino acids. So normally each protein is composed of a chain of 20 amino acids. Okay. By the way, there is a reason why there are 20 amino acids. Apurv Patel from ISC Bangalore, he has shown once that there are quantum algorithms back in DNA, right? So the quantum biology, which is another important area. So why we have 4 nucleotides, ACGT, and 20 amino acids? Because we are nature is using quantum algorithms. And I think people have shown that the migrating birds use quantum algorithms to find the destination. So nature is already using these kind of quantum computing. Okay, yeah, right. So all these things are connected. Okay, in 1902, Emil actually showed amino acids are linked to form protein, right? So this is something, a chain of amino acids will form a protein. What is amino acid? I will tell you in details, right? I'll show you those things. So in 1911, Thomas Hunt Morgan discovers genes on chromosome are the discrete units of heredity. So that means they observe that genes are transformed from one generation to the next generation. Okay, this is actually genes. The copy of gene goes from one. So actually, in any child, they get one copy from the father, one from mother, right? And that goes on. Okay. RNA was discovered in 1911. RNA is a beautiful molecule. I love RNA because RNA. People say that in the beginning, origin of life, if you read some of the books and some of the claims, people say that on the planet, first RNA was created. Okay. And before RNA, there was a couple of other chemicals and they formed RNA. But RNA is highly unstable molecule. Okay. It does have information, but it's highly unstable. So nature discovered DNA to store data. Nature is actually storing all the data instructions in DNA itself. Okay, because that is more stable. That is how this happens. Now you see 1940 to 1950, I think we got independence in this time, right? Then in 1941, actually these two people identify that genes make proteins. Something interesting thing. And this complementary nature that we use is only appeared in 1950. See the stage at which biology was when actually 1950 already Hamming error correcting code was there, right? But still, how far we are in biology? We just learned that A complement is C and G, right? And T complement is A. Those kind of things were observed only in 1950. tRNA. tRNA is an important molecule known as transfer RNA. So when actually what happens, the entire biology, if I ask you this, what is the summary of biology? There is only one process which is known as central dogma. What central dogma tells? Information is stored in DNA because it stores all the information on an instruction set for making us who we are, right? Or even for any living organism, right? Now this DNA is converted into RNA, okay, especially messenger RNA, and that messenger mRNA, they call it messenger RNA because it is taking message from DNA to to give you to the assembly of proteins, right? The the amino acids. DNA to RNA, RNA to protein. This is how the information flows. This is known as central dogma. Every time your body wants to make some protein, central dogma happens. Okay. DNA to RNA, RNA to protein. Now, this is very simple version of it, but there are many other things which are involved in this process. tRNA is one of them, which helps taking one bit of RNA and attach one amino acid to form protein. Okay, I will show you a video also. It is available. You can see how this happens. Okay, so this is something really very, very important. Okay, then comes the year 1953. Watson and Crick reduced the double helical structure of DNA. This is the most important discovery that people say in biology. Double helix structure was discovered by these people, right? So DNA is there. Now I think, as you know, there are, I mean, yeah, you may not be knowing, but I'm saying that you will observe that whatever you do in your life means when you interact with the environment, any species, certain things are changing all the time in your genome, and that is called as epigenome. So, for example, if you are smoking, what happens if you smoke? If you smoke, methylation frequency, CS3 molecule will increase. Okay, we call it hypermethylation inside the genome, and that transfers to next generation. Now what happens? You may not have any problem, right? You smoke, no problem. But then maybe third generation or fourth generation, somebody will have lung cancer. Karma ka phal, transferred. Close to Gita, right? So this happened, people have now identified that. So that gene is expressed, right? That, and the faulty things are produced because, the genome has been altered, the amino, epigenome has changed. So epigenome not only involves actually change of these quantity of these molecules like CS3 and everything, it also the structure changes. So many things are changing. Every time you are interacting with the environment. So tum jo kuch bhi karm kar rahe ho, it is recorded inside your body all the time. This is proven, right? So that means whatever you do, so next generation are going to affect. So ab samajh rahe ho na? Cigarette to band kar diya? Yes, that's what I'm saying. So your next generation, third or fourth generation may not be smoking at all. मैंने तो कुछ किया ही नहीं है, and then you got lung cancer. पूर्व लोगों का कर्म ट्रांसफर हो गया, right? So this is something very powerful idea actually, right? तो इसी हमारे गीता में तो ये सब कहा ही गया है, right? कर्म का सिद्धांत से कोई बचा नहीं है, right? भगवान भी नहीं बचे हैं, तो basically it's very very so when you look at biology, you actually see the things, how things are happening. And epigenetics has started very recently. Okay, so now people can correlate the things, okay, right? There's a lot of epigenetic data has been discovered, and it is there already on the websites. You can analyze it. Okay, so David Baltimore is okay. He's another very important. He was a president of Caltech. I have interacted with him some time back actually. He isolated the first restriction enzyme. Restriction enzyme is an enzyme basically made of proteins basically. Restriction enzyme acts as a scissor which can cut DNA at a specific location. Okay. So actually these restriction enzymes are quite important. If you want to cut the DNA at a specific location, you should know what restriction enzyme to use. Now there are softwares actually, which you can use to cut the DNA at a specific location and do editing. By the way, as you know that the most dangerous, or I would say the most disruptive thing that has been discovered ever by the mankind is CRISPR. Jennifer Doudna at UC Berkeley, she was responsible and she got the Nobel Prize also. So CRISPR is a very, very important technology which allows you to easily modify the genome. So you can edit the genome. You can switch off a gene, switch on a gene, right, at your wish easily. Okay. And you can do modifications. So that's CRISPR is really very, very powerful. And of course, obviously government wants control on it also, right? You should not, all kind of things are happening. Okay. But CRISPR you should know and read about CRISPR. It's a very, very dangerous technology. And okay, so in DNA storage also people are using CRISPR for storing data. It's something very powerful technique. David Baltimore also classified HIV viruses, so a lot of work is there. I think. Then you see, 1986, Larry Hood, he was very famous in systems biology. He developed automated sequencing mechanism, right? So when you do manually everything, it's a very challenging thing. So people started making robotic things and so on, which will do automatic, sequencing means reading the data. So you see, 86 Human Genome Initiative announced in 86, right? Remember, as I mentioned. So in 1990, the 15-year Human Genome Project was launched by the US Congress. The US government launched that. And then 1995, actually this is the copy which I saw. Chromosome number 22 was published, I think in September or December, I forget. Still have the copy with me, by the way, from the Nature, that was published. Craig Venter, I remember, he was in the team of Eric Lander. The first bacterial genome was sequenced in 1995. Fluorescent sequencing method was often eukaryotic genome of yeast. Yeast is a very, very interesting thing. Yeast तुमने देखा होगा, right? खमीर ना जो bread बनाने के लिए use करते हैं, right? Yeast. Yeast is at some point of time, yeast was the first thing which people know the entire proteome. Proteome means all proteins that are there. Whole structure was known to humans now. Okay, this is something really important. In 1996, E. coli bacteria was sequenced in 1997. So complete first human chromosome number 22 was sequenced in 1999. And finally in 2003, Human Genome Project completed, right? So now after that, lot more. That's why a lot more things are happening. Yeah, that's all I think. So I'll stop at this point. Right, next class, I'm going to talk a little bit more about these biological things, right? So that you have some good understanding what is happening. Okay, thank you. By the way, you got the next set of videos for next quiz. So quiz may happen. आज तो नहीं, अगली बार में तो होगी, right?